Hopefully now. I think it's working. Yeah, possibly. I will introduce the speaker first of all, so let me share my screen for a second. So this is the first meeting of the year, the Population Dynamics Virtual Seminar. First of all, I would like to say that we have a new website and a new Zoom link. I know that there are problems with the newsletter, so if someone received newsletter in the spam, let me know. Today we have Bartlomiej Waclaw from the University of Edinburgh, and actually has also an affiliation in Poland now. Then he will present a talk about substrate geometry affects population dynamics in a bacterial biofilm. A few words regarding Bartlomiej. So he studied statistical and biological physics, and he has a focus on microbial evolution, bacterial colonies, and cancer. That he studies with computer simulation, analytic calculation, and wet lab, basically a spectrum of everything. And he got a PhD in theoretical physics in Poland. Then he moved for a postdoc in Germany, Leipzig, and then for again another postdoc in the UK, Edinburgh. Well, he got a reader position, and now he's also leader of a new center, the Oscury, in Warsaw. So with that one, I would like to leave the stage to Bartek. So probably you can start sharing the screen, and it's yours. Thanks.
Thanks. Can you see my screen?
Yeah, full screen, perfectly.
Yes.
Thanks.
Yes, it's okay. Thank you, Marco, for this nice introduction. Yeah, as Marco said, I now share my time between Warsaw and Edinburgh. And actually, what I'm going to tell you about today has been all carried out in Warsaw in this new group called the Oscury Center for Physics and Chemistry of Bacteria. I'm very happy to have been invited to this population dynamics seminar series, because it's something that Rosalind initiated, and Rosalind was also the person who somehow introduced me properly to biological physics, because I was toying with the idea of switching from statistical mechanics to biological physics for a while, but somehow never got the chance to really do it. And then when I arrived at Edinburgh, Rosalind was there, and she told me what it is about, and we started working on some projects together. And we've continued since then. Okay, so maybe what I should do first is to actually tell you about who is involved in this project that I'm going to discuss. So my screen froze. Hopefully it will. Okay, it's working again. So okay, let me just do one thing, because I can't see my slides properly. Hide the floating meeting controls. Okay, so so the people involved in this project are Witek Postek, who is now at Broad in Boston, Claudia, who is a PhD student in my group, and Ellen Lilia, who doesn't want her picture to be shown. She's a postdoc, and this is me showing some kids how to use microfluidics to kill bacteria at a science fair. So before I go into the main part of this talk, I wanted to briefly introduce the general idea of studying population dynamics in biofilms. I don't know why my computer is lagging behind so much. I'm trying to swap. Okay, maybe I should just start this presentation again and see what happens. It's not much faster now. Anyway, so I would like to first introduce a little bit what we do in general in this new group in Warsaw, and also the biophysics of biofilms and population dynamics of biofilms, because I'm not sure how many of you are familiar with these kind of problems. So our research group in Warsaw is mostly interested in studying experimental and theoretical models of bacterial growth and evolution, in particular in vitro models of bacterial infections. So for example, something that we can set up in a microfluidic chip. we also do evolution of resistance to antibiotics. That's something that we started together with Rosalind in Edinburgh when we were both there. And growth and evolution of bacterial colonies and biofilms. And this is what I will tell you about today. Okay. Okay. Move to the next slide. Okay. So to those of you who don't know what microbial colonies and biofilms are, it's generally anything that grows on the surface and is made of bacteria. So colonies in a petri dish, which those of you who have some microbiology experience must have done, they qualify as colonies or biofilms. You can also have in vivo biofilms in the form of some bacterial conglomerates that grow, for example, in the lungs of cystic fibrosis patients, which is shown here in this picture on the right. You can have bacterial colonies growing inside animal or human epithelial cells, which is shown here at the bottom. And you can also have biofilms or colonies of bacteria growing inside medical devices. Like, for example, this is a section through a urinary catheter in which there is a thick biofilm, which is in addition encrustated with some mineral deposits, which makes it a very complex structure. So in my research, I've been trying to understand how physics affects the growth and evolution of biofilms. And this is because I'm a physicist, so this kind of angle is somehow, it comes natural to me. And what we have done over the last 10 or so years is to look at, for example, how the shape or rigidity or adhesion between bacterial cells affect how bacterial colonies grow, and also how various physics or chemistry interactions with the environment affect this process, including diffusion or nutrient uptake and so on. And this used to be quite a niche topic, I would say. However, now I think it has percolated into mainstream biology, at least judging by the fact that this review appeared in Cell a few years ago. So I think this is now more commonly accepted that physical or mechanical interactions are important for bacterial growth and especially for biofilm formation and evolution. Okay, so what I specifically mean by biological evolution in biofilms is something that you can actually observe yourself if you go to the lab and do a simple experiment growing some bacterial colonies on agarose. You will sometimes see that instead of forming nice and round colonies, some protrusions develop emerging from these colonies. And these usually be some new genetic or epigenetic variants that grow a little bit faster or spread a little bit faster on agarose. And this can also happen in biofilms that occur in nature, not only in the lab. And such new variants are often undesired because they can, for example, be resistant to antibiotics or have increased pathogenicity, or maybe they will interfere with some industrial processes in which biofilms are important. So what interests me is to look at what factors determine whether a mutation that has newly occurred in a biofilm forms such a macroscopic sector or actually takes over the entire population, which can also happen. By the way, if you have any questions, you can just interrupt me and ask the question, and do it orally because I don't think I can see the chat. Okay. So.
Sorry, right on cue, I have a question.
Yes.
Yeah. So when you talk about evolution on biofilms or mutants appearing on biofilms, is it because of the structure of biofilm, or is it just because a lot of cell division happens at the interface?
Yeah, a lot of cell division happens. So
Okay.
the biofilm or a bacterial colony must be large enough for such mutations to occur. How large depends on the mutation rate. this in turn depends on what is actually that is going to be selected in this biofilm, because it might be that many different mutations give the same sort of phenotype that will be selected and that will cause this expanding sector to grow. Okay?
Yeah, thank you.
Any more questions? If not, I will then continue. So I decided to give you a small biofilm bacterial colony population dynamics primer because I wasn't sure who was actually going to listen to this talk. So apologies to those people who know all of this already. So so regarding these mutant sectors that emerge in bacterial biofilms, a mutant that occurs spontaneously does not always have to do that. So this phenomenon in which a mutant forms a macroscopic sector, which is demonstrated in this video from a computer simulation here. I hope you can see the video. So this phenomenon only occurs if this mutant somehow manages to stay close to the expanding edge of the biofilm, because this is where nutrients are and this is where the mutant can replicate, and only then it can form these microscopic sectors. In most cases, what will actually happen is what is shown here in this panel, which says extinction. If the mutant somehow lags behind the growing layer of the biofilm, then it gets trapped in the bulk of the population and forms only a very small sector, which might not be really visible. So that's one thing to have in mind when I come to this main part of the talk about substrate geometry affecting this process. The other thing to take into account is what happens when there are many clones or many variants at the same time, because usually they will be in a large enough colony and they will somehow interfere with each other. The simplest thing to do to understand what happens in this case is to consider only two clones, which are identical, save the color. And this is a result from a computer model which simulates an expanding colony, and the camera follows always the trailing edge of the colony. So only this part of the colony where the actual growth happens. And you can see that these red and green sectors formed by the two clones, they sort of grow or shrink depending on the lag, basically, because there is no asymmetry here in the growth rate. And eventually, if we wait long enough, only very few of these clonal sectors will survive. Most smaller sectors will collapse, and larger sectors will grow at the expense of the smaller ones. And this is nothing else but just genetic drift, this term from population dynamics, which describes random variations in the frequency of genetic alleles or genetic variants, more generally speaking. So that's another thing to remember. And the last thing is what happens when one of the clones grows a little bit faster than the other one. So here, for a change, I show some experimental results in which the yellow clone in a bacterial colony has a fitness advantage over the blue clone. And if this fitness advantage is very small, or actually when there is a small selective disadvantage, the yellow clone, even though it initially has a very high frequency in the population, because it's been started with a mixture of the two clones put together on a petri dish, and then they started to grow into this colony. So for a small selective disadvantage, the yellow clone forms very tiny and very few sectors. But if you increase its selective advantage, meaning that it begins to grow faster and faster, then these sectors become larger and more of them appear, and eventually you can get to the point when they completely surround the less fit genotype. And this is just the demonstration of natural selection in the biofilm. So is this all clear, or are there any questions? Okay, if there are no questions, then I will proceed. So last thing before I go into the main part of this talk is what we have actually learned about what determines the strength of genetic drift and the strength of selection in biofilms from the physics or mechanics point of view. So one thing is which is quite important is even things that you would normally not consider to be important, like the cell shape, can determine the fate of a fitter mutant. In this case, it was a mutant resistant to an antibiotic. For round cells, this mutant will produce many more sectors than if the cell is rod-shaped. And this is down to some interesting mechanical interactions that happen in the biofilm or the colony between these cells. Another interesting observation is that even in the absence of growth advantage, a mutant might still form these expanding sectors simply because it adheres less to the substrate on which the biofilm or the colony lives. So this has nothing to do with growth advantage. It's a selective advantage, but it's related to mechanical interactions. And lastly, other things like friction anisotropy, which has to do with the cell shape again, whether it's equally easy to move or equally difficult to move the cell along its major axis if it's a rod or perpendicular to it, that can also matter a lot. And this is shown here in this computer simulation in which we investigated two different types of cells with isotropic friction, meaning that was pushing the cell in every direction was equally hard or equally easy. And for a round shaped particle, it was much easier to push it along the main axis. And then starting from many different alleles, many different mutants, you can see that there is a very large difference in the number of sectors or clonal sectors formed in this simulation. Okay. So let me now go to the main part of this talk, which is
sorry.
yes.
Can I ask a question regarding this red and green clones which you showed? So as I understood, the starting was they were like more mixed clones with small sectors only, and as with time they became like more demixed kind of.
Yes,
so
yes.
Is there was it only like genetic reason, or is there any physical reason for this change?
This demixing of sectors is purely statistical in its origin. And I think this simulation shows it. So if there are small sectors to start with, randomness due to cell replication and sometimes maybe cell death and also cell movement cause some of the sectors to fluctuate, or all of the sectors to fluctuate in size, and some of them just fluctuate down to zero. And then the two sectors that are around such a small sector will collide together and form a larger sector. So eventually this leads to the formation of larger sectors. But in the case of a radially expanding colony, there is also this fictitious force due to the colony's perimeter expanding, which eventually moves these sectors apart to the point where they cannot collapse. So if the sector is large enough to start with to survive until the colony is large enough, then it's more likely to survive.
Okay, thanks.
Any more questions? Okay, so let me move on. All of this research that I have presented and also most of the research done by others have been done in the lab on flat surfaces, either on agarose petri dishes or small pieces of agarose under the microscope, or things like flow cells for culturing biofilms. But in nature, it's often not like that. Biofilms grow on all sorts of surfaces that can be rough, not flat. And so the natural question is, how is population dynamics and all these things that I've just presented affected by surface roughness? So this is what we wanted to investigate, but of course we do basic research. We don't go and look for biofilms in the nature. So we've designed an experiment that can be carried out in the lab in a microfluidic chip that explores the possibility of having a corrugated surface on which the biofilm grow. And so the idea is to create a small compartment with a corrugated surface with varying amplitude and the period of corrugation, inoculated with bacteria, let's say only two types of bacteria, red and green, which differ only in the color, and see what happens. So and then, of course, to see what happens, we have to somehow image it, and we image it under the microscope. And this is how a picture from taken in the bright field mode looks like of these biofilms when they eventually form in the microfluidic channel. I will describe this channel or this device in more details very soon. Just to mention that for this to work, you have to use a bacterial strain that sort of forms biofilms, and we've used this particular strain which has been isolated from urine. So, but it's not uropathogenic. It's actually it's been actually used to eliminate uropathogenic bacteria in some medical trials. And we've modified it genetically so that we could have red and green fluorescent versions of this strain, and also some variants resistant to the antibiotic rifampicin, which will be important in the later part of this talk. And this is the actual microfluidic device. It's a very long channel which supplies the growth medium to the biofilms, but all these biofilms grow in these much smaller side pockets or side wells with different levels of corrugation. So this is how it looks when we zoom into one section of this channel. So through this main channel, there will be always some bacterial medium flowing, and biofilms will grow only in these small side pockets. And if there is any growth happening in the main channel, that's not good, because it can use up the medium and cause problems to the biofilms in the side pockets. So what we do is we just flush this channel periodically with a very strong flow rate, and this eliminates anything that could attach to the main channel and grow here. So we only end up with bacterial biofilms growing in these little wells on the sides. Okay, so I will show you now a video of what happens when this system is inoculated with two strains of E. coli. One is green, one is red fluorescent. They are initially in equal proportions, and then we let them grow for a couple of days. And what you will see is that eventually these sort of clonal sectors form in all the wells of different shapes, but where the sector is actually positioned depends on whether the surface to which they attach is corrugated or not. So in the case of a flat surface, these sectors form at random locations, but in the case of a corrugated surface, the sectors form preferentially in the grooves at on the surface to which the biofilm attaches. So maybe just to make it clear, what we see here is a kind of a two-dimensional biofilm, right? So it's not a single layer of cells, but it's a relatively thin biofilm, so that you can see into it to a certain degree. But we are not using high magnifications here to see individual cells. We are just looking at the overall population, sort of. Okay, so then we can also make this a little bit more quantitative and see. So if what we observed in this video was correct, and indeed the clonal sectors formed preferentially in the grooves, then if we plotted the position of sectors boundaries, they should somehow align with the positions of the grooves. Of course, you can see already from this picture that it's not going to be exact, because these biofilms, these sectors bend a little bit, but there should be some correlation between the positions of the grooves and the positions of the boundaries. So this is what we do here. So we plot the probability distribution of finding a sector boundary at a particular point across the well, where this position is just some normalized position so that it varies from 0 on one side of the well to 1 on the other side of the well. And you can see that there is indeed a very high degree of alignment between what would be expected on the positions of the grooves and where actual sector boundaries are. And this happens in all these cases with the grooves, but not in flat-bottomed wells, where you basically get some random distribution. Although it's not a flat distribution, which is also interesting. Okay, so this is another way of showing the same thing, but now in time. So these are sort of chemographs now showing how this probability density of finding a sector boundary at a particular position changes also in time. And you can see that these sectors form very quickly. You know, after sort of 20 to 40 hours, they will already be quite well developed sectors, and then they just continue to stay where they were. Okay, so we would like to understand a little bit why this happens. Why, for example, these sectors do not spread the way they did in bacterial colonies growing on agarose, and why they do not merge this much. So to understand this, what we did was to determine the velocity field of a growing biofilm. So what's the velocity of a small patch of a biofilm at any point in this well? And we did this using the method of optical flow, which is based on taking very short videos of the growing biofilm. So this is slightly sped up. I mean, this whole thing would normally take only a minute. And you can see, if you carefully look at it, that during this time, the biofilm moves a little bit due to growth, but it's not a lot. But this method of optical flow is extremely sensitive to very small amount of movement, and it can be used to determine how the biofilm moves. And from this, we can, for example, calculate. Yes.
What do you actually do? Do you like subtract the two frames from each other or something?
No. So it's a little bit more complicated. So the assumption is that when you go to the next time point, the image will look almost the same plus a small correction. And this correction will be the image from the previous plane propagated by the velocity field in the biofilm. So you have to somehow calculate how the optical density of the image will be affected by the velocity field. And actually, it might sound complicated, but it eventually leads to a linear equation that needs to be solved for this velocity field given the two frames at time t and t+1.
Oh, that's nice.
Mm-hmm. So from this, we can determine this velocity field and show that in our experiment, first of all, this velocity field is almost vertical, meaning that the biofilm grows mostly towards the opening, towards this side which is open to the main channel and where the surplus of the biofilm goes and is removed, but also that the growth rate, which can also be determined at the same time from this method, does not really change very much. So these biofilms are sufficiently thin to grow almost exponentially and with the same rate everywhere. And this is not really crucial for what comes next, but somehow it simplifies modeling this system, because we also want to have a computer model that reproduces these results. So when we
like if you wanted a more kind of realistic biofilm with a modulation of growth rate, could you just make the things longer?
Yes, and we did that, and it seems that for this specific type of bacterium and then the medium and the temperature, if you go below approximately 200 microns, so if the biofilm is thicker than 200 microns, then growth basically stops at the bottom. Yeah. So it doesn't really make sense to go much deeper than this. But also this what I'm going to say about this sectors forming in the groups, that would also apply to taller wells or deeper wells, as long as there's growth at the bottom. because only then the biofilm sort of feels these corrugations at the bottom. Otherwise, they are not relevant at all.
Thanks.
So we can, yeah. So as I said, we can show that the velocity is vertical, and the vertical component of the velocity increases almost linearly with the distance from the bottom, which is what one would expect from an exponentially growing biofilm in the entire volume. So one thing that I should maybe stress, and this is different to other types of biofilm experiments, is that we make these biofilms grow in these indentations, these little wells, and then they are constantly supplied with fresh medium from the top. And they cannot grow thicker than 100 microns, which is the depth of this well. If you have a naturally occurring biofilm that grows on a flat surface, for example, and the flow rate is not very high, this biofilm can grow thicker and thicker and thicker, and eventually the bottom of the biofilm will not grow. So this is not this regime. This is more like, if you think about where this would be relevant, is more like biofilms growing into little cracks in a pipe or something like that, but not clogging the pipe. Okay, so we know that this velocity field is sort of aligned with side wells of side edges of the well, and now we wanted to see what actually happens at the bottom, because it's the bottom that seems to be important for determining the dynamics of these clonal sectors. So it turns out that the only appreciable amount of movement in the horizontal direction actually happens at the bottom. So when we measure the horizontal component this time, it was very high at the bottom, but there was very little movement away from it. But then when you think about these wells, these corrugations are actually at the bottom, so they will arrest this horizontal movement exactly where it happens when the amplitude of these corrugations is large enough, and then this prevents all these sectors from mixing, because there is almost no lateral movement. So these sectors just propagate in the vertical direction due to growth, but they do not mix. They could mix only at the bottom, and this is what happens in a flat-bottomed well, but not with the corrugated well. Okay, so this is also somehow related in an interesting way to the phenomenon called buckling, which has been studied in bacterial colonies. Rosalind and myself and Pietro Cikuta and Matt also wrote a paper about it, but then some other people also published on this. So it's basically when you have a surface and you have some bacteria attached to the surface. In our case, it will be the bottom surface where the biofilm starts. And when these bacteria begin to grow, they will push on each other. And then even though this bottom surface, if it's flat, it would try to align them in a line, this pushing will eventually cause some of the bacteria to pop out of this aligned layer of bacteria and stand vertically. So it will be something like this. Even though there is this alignment initially and some compression coming from the growing biofilm, some bacteria will pop out due to growth and this buckling phenomenon, and they will change their orientation. And we've actually discovered that in this case of our experiment, such buckling is it occurs, and this is the source of this movement. This buckling is suppressed, and this is what causes a lot of movement at the bottom. And to understand this, it's good to look at this simulation of a growing biofilm, which starts from a similar configuration to what we do in the experiment. And you can see here that, for example, this red sector that has formed, it sort of moves around at the bottom. And so this is what causes sectors to merge in the case of flat-bottomed wells. But this wouldn't happen if we, for example, used wells with lower height, because then what would happen is there would be less compression and this buckling would be easier to occur. And then cells would naturally, very quickly orient themselves vertically, and then there would be very little movement at the bottom. And we actually see something like this when we increase bacterial adhesion to the bottom by treating the surface in a certain way. Then this makes the cells to really align more in this way, even if we do not change the thickness of the biofilm. So you can achieve this basically in many different ways, but the reason why this movement occurs in the first place is this suppressed buckling. Anyway, so the last part of this talk about what happens is what happens when one of the strain has a selective advantage. So suppose that we again start from a mixture of red and green bacteria, but the red strain this time grows a little bit faster, 50% faster than the green strain. You will see that it will form sectors that now expand over time, and eventually, when we wait long enough, this red strain will almost completely eliminate the green less fit strain. I mean, I will maybe speed this up a little bit. You can see that eventually we get rid of almost all green bacteria. So this is what happens in a flat-bottomed well, okay? But it turns out that this is not what will happen in corrugated bottom wells, because these corrugations arrest the spread of the fitter variant. And even though the fitter variant can grow much, much faster in principle than the less fit variant, it will not be able to spread because, as I said, in order to do this, it has to somehow move around at the bottom. But if this bottom, if the movement of the bottom is suppressed by the grooves, it cannot do that. So this will lead to the coexistence of fitter and less fit strains or clones in the same well, which would normally not happen, and only in the flat-bottomed well, only the fittest clone would survive. So this is what our computer model would predict, but it's also actually what happens in reality when we do an experiment like this. So in the experiment, we start with two strains which have the same fitness, but one of the strains, the red one, has a mutation which makes it resistant to rifampicin. And this mutation doesn't do much when there is no rifampicin in the environment. So when we just start this experiment, the red strain, which is initially much less abundant, it forms tiny sectors, but these sectors do not really expand very much until when we start adding rifampicin, because this gives the resistant strain a selective advantage. And then at some point, when we add a lot of rifampicin, these sectors begin to expand rapidly and eliminate the green sensitive strain in flat-bottomed wells, but not in the wells in which there are some corrugations. And when we remove rifampicin again, then the sensitive strain comes back in these corrugated wells. So there is a qualitative difference between a flat-bottomed and a corrugated well. And of course, there is some transition between this and the corrugated well, depending on the amplitude of corrugation. So these corrugations have to be sufficiently deep, but as you can see, they don't have to be very, very deep for this to occur. Okay, so we can also model this using the computer model, which we feed with this time with the parameters like the growth rate and the growth time and rifampicin treatment as in the experiment, and the model can actually predict what will be the resistant what will be the mean clonal fraction of the sensitive strain at the end of this experiment. And this model has several variants. One of them has a lot of adhesion added to the model, and this somehow doesn't work that well. So it means that adhesion is somehow not very important in this phenomenon. Okay, so that's the end of my talk. So the conclusion from our experiments and simulations is that corrugated surfaces, first of all, reduce genetic drift, meaning that there is less mixing between different clones that occur in a biofilm. And then they also reduce selection, which is somehow even more interesting, meaning that a fitter strain does not necessarily invade the less fit one in the presence of this corrugation. So in principle, one could imagine using these corrugated surfaces to control population dynamics in bacterial biofilms. Now, how this is relevant for things like, you know, industrial processes or medicine, I do not really want to speculate that much. I think it's an interesting basic science phenomenon that needs to be further explored, and maybe it could find some applications, but I think it's a little bit too early for that. So that's the end. I would like to thank you for your attention, and also thank you my sponsors and my team, especially these 3 people who really contributed to that work. And that's it. So thank you very much. If there are any questions, I'm happy to answer them now.
So thank you, Bartek, for a very nice talk, very interesting. From this moment, I will stop the recording.